Welcome everybody to the Woman in Optics series webinar. So my name is Larry Teran Suarez. I am the president of the Optical Student Chapter at the University of Puerto Rico. And with me today is the Vice President, Diego Gutierrez. We are gonna be co-hosting this meeting today. And I hope that you will enjoy and take advantage of all the information that we are prepared for you today in this meeting. So with that being said, let me start with our first speaker, which is Alessandra Carmichael. She is originally from Cadiz, Spain, and she is currently a postdoctoral fellow at Burns Lab and Indiana University School of Optometry. So Alessandra has a background in physics from the University of Sevilla, Spain, and she has spent a year in the University of Münster, Germany. And she is right now an Optica Ambassador of 2022. So welcome, Alessandra.
Thank you very much. Let me share my screen. Can everybody see my screen correctly?
Yes.
Yes, now we can see it.
Okay, good. So thank you very much for the introduction, and also to the University of Puerto Rico Optica chapter in general for putting this series together. I think you've done fantastic work of getting people together and advertising and everything. So congratulations to you on that too. So as you just mentioned, thank you very much for the introduction. My name is Alessandra Carmichael-Martins. I'm currently a postdoc at the University of Indiana in Bloomington. And today I'm going to talk about optical systems for human ophthalmic applications. So though you did cover a little bit of my background, I'm going to go over that a little bit more. So as you mentioned, I did my bachelor's in physics from the University of Sevilla, but I decided that I wanted to specialize in optics and learn a little bit more about it. So I decided to take a year abroad in Münster, Germany, where I was actually able to get much more experience on hands-on work, in particular with optical fibers. And I still didn't want to go back to Sevilla because they didn't have optics. So I decided to take another year with a scholarship to the Complutense University of Madrid, where I continued doing several elective subjects in optics. And in particular, I did my final year project on holographic filters applied to renewable energy. Wanting to continue in optics, but not knowing quite how to do that, I came back to Sevilla to do an internship at the Spanish Research Center in the Institute of Microelectronics, and I worked on characterizing silicate photomultipliers and avalanche diodes. But I realized that even though I really liked this, if I didn't continue my studies, I wouldn't really be able to get into research. So I went on to do my master's at Imperial College London, where I worked on several projects, and I loved my experience there, working on optical trapping. But I've specifically specialized in optical design and manufacturing of a high numerical aperture lens for fluorescence microscope. So this was done in conjunction with a company called Orox, which actually made the fluorescence microscope, and my design was actually implemented in some of their newer, later designs. I then decided that I wanted to apply my knowledge in optical design to the medical field. So I decided to go do my PhD in Ireland, in Dublin. So I decided to work on more ophthalmic applications under the supervision of Dr. Brian Bonson, and I'll talk a little bit about my work there in a few minutes. During this time, I also did research visits at the Spanish Research Center in Madrid at the Institute of Optics, and also another month at the Laboratory of Optics at the University of Murcia. During this time is actually when I got involved in Optica in general, and I was the president of my student chapter. We organized several events to invite new people to join our chapter because it was quite small at the time, and invested a lot of time in outreach events too. So this in particular was one that was organized as a UCD festival, which attracted over 10,000 people. And even though it was a lot of work, we did have a lot of fun organizing these events for kids. I was also one of the main organizers for the IONS Ireland conference in 2021. Unfortunately, this was affected by the pandemic and we had to switch to a virtual format. But luckily, I had an amazing team with Kevin Shortis and Andrea Pacheco, that between the three of us, we worked hard, but actually worked very well together and were able to have an excellent experience. And even though it was virtual, we were able to actually get all our winners for our best posters and best talks to come over to Ireland for an entire week, fully paid for, in 2022. And we were able to do research visits to different optics labs in Ireland, including industry as well. So now I'm going to move on to talk a little bit about my PhD, which was funded by the Horizon 2020 program, and it was called MyFun. So this was Myopia Fundamental Understanding Needed. And it was a consortium of 14 different PhD students. It involved 5 different countries and 5 different academic institutions and 2 companies that are down here. And we all worked together towards the further understanding of myopia. The reason why we wanted to look into myopia and what myopia is basically in normal viewing conditions, we have light entering our eye and focusing on the retina. However, if you're myopic, light tends to focus in front of the retina and hence causing a blurry image. And as most of us may know, you can correct that by using a minus lens in front of the eye. But the bigger problem is understanding what is actually causing this, because currently in world population, about 50% of the world population is estimated to be myopic by 2050, and no one has been able to actually stop this or understand the procedures in order to be able to create a solution for this problem. And even more, the high myopes are actually increasing as well, which means that they are more prone to other diseases. So this is why the whole project was put together. And my thesis in particular revolved around digital micromirror devices. So a digital micromirror device is an array of micromirrors that can take two states, either an on position, which reflects light in one direction, or an off position, which would deflect light towards another direction. And it can only be that binary option. In this case, it had a very high fill factor of 92%, and I in particular was using it at 22.7 kilohertz at the time, which was the maximum, but now with the newer updates, you can actually go up to 50 kilohertz. So now to explain a little bit about the eye, as we know, light enters through the center of through the front of the eye, goes through the cornea and gets focused onto the retina, onto the back of the eye by the effects of the cornea and the lens in the eye. The back of the eye, or the retina, is actually a complicated structure, which I won't get into in detail, but if we look at this image, we know that light enters through the bottom of this image here, travels through several layers that are mostly invisible or transparent to our imaging techniques, and then gets absorbed by the photoreceptors that are located towards the back of the eye. There are 2 different types of photoreceptors that absorb light. Those are rods and cones. Rods are in charge of dark viewing conditions, so for example, at night vision, and cones are in charge of day vision and color vision as well. So there are 3 types of cones that absorb short wavelength, medium wavelength, and long wavelength. And the part of the photoreceptors that actually absorb light are the outer segments, which I'll talk about on the next slide. In this case, what my project, one of the applications of my project was actually characterizing what is known as retinal directionality, or the Stiles-Crawford effect. So basically, if light enters through the center of the pupil, we see a certain brightness. But if light, the exact same light, enters towards the periphery of the pupil, we start seeing a darkening effect. And we don't really understand what the cause of this is. There are several theories out there, including wave guiding within these sections of the photoreceptors, but that lacks a lot of details and specifications to fully explain it. So what I did was build a system using a digital micromirror device to be able to characterize this effect. So we used the DMD to select a small portion of the beam, project that onto the pupil plane, and the subject was able to compare between the reference, which would be the center beam, and any of the test beams that scanned the pupil, getting similar results to what is shown here. The biggest part of this was actually coming up with a theory that could explain all the effects. So what we did was come up with a very simple approach, which is taking into account the overlap of the incident beam, in this case, blue light, for example, with the outer segment of the cone that absorbs that light. So in this case, the blue cones would absorb blue light, the green cones would absorb the green light, and so on. So by doing this in a very simple overlapping approach, by modeling in COMSOL both the incident light and the outer segment, we were able to get curves that resemble what we obtained experimentally, but also explain several effects that other theories could not. For example, when several of these rods of these outer segments are grouped together, we get a lessening effect, so a flatter curve, which would explain why we don't get such a high effect in rods, for example, that tend to be highly grouped together. The next part of my application was actually designing a new wavefront sensor. So typically we use Harman-Schach wavefront sensors, which for ophthalmic applications, which I won't explain here, but they have some limitations on measuring very high aberrations. So what I did was design a new system where I use near-infrared light to focus light onto the retina, and we use the back scattered light from the retina to focus the light, to image the light coming out of the pupil of the eye onto our DMD sensor. In this case, we do have a Harman-Schach put in the system, but that is solely for comparison purposes. Like this, we can sequentially scan our beam and in different regions and use the full amount of our CCD detector to be able to capture the point spread functions. Each one of these point spread functions will have a particular location that we can compare to a non-aberrated beam. So by estimating the measured displacements for each one of those sectors between the aberrated beam and the non-aberrated beam, we can actually reconstruct the wavefront that we're looking for. This allows us to measure much higher aberrations. So this is an example here where this wavefront sensor worked and the gold standard Harman-Schach did not. And an example where I'm showing an actual real case of a high myopic subject where we can actually measure their aberrations, also without the limitations that the typical Harman-Schach has. So this was more or less everything linked to myopia. And most of these were, so especially the first part, were psychophysical measurements, but I wanted to continue on into more medical applications and particularly high resolution imaging. So I decided to cross the pond over to the US and do my postdoc in ocular imaging with Dr. Steve Burns. During this time, I've actually been able to be a PI for two of my current grants. And I was also selected as a 2022 ambassador with the rest of my class. And it's probably the reason why I'm here today talking to you in the first place. So at Frontiers this year, where I was able to meet the rest of my class and several of the other ambassadors, I was able to talk to several of the students and start setting up some of the projects that are going to be moving forward over the next year. And it's honestly been an amazing experience. So during my postdoc, actually, I've been working on two different projects, which I'm going to explain here. The first one is high resolution imaging of the ocular drainage system. So what we want to do with this project is actually image the trabecular meshwork structures in a new manner, which I'll explain in a minute, but also to design a new lens to be able to improve on this type of imaging. And the second part of this talk will be adaptive optics imaging, which I'll get to. So to be able to talk about the ocular drainage system, I first have to talk about glaucoma. Glaucoma is the leading cause of irreversible vision loss and blindness in the world. It affects 64.3 million people worldwide, but its cause and progression are poorly understood, and currently the only modifiable risk factor is intraocular pressure. So basically, the front of the eye, and particularly the ciliary body, creates fluid in the eye that flows around the iris, through the front of the eye, through the pupil, and back out, and is filtered and exits the eye through the trabecular meshwork. However, if the trabecular meshwork gets occluded, this would increase intraocular pressure, and hence why we want to look at this region. So it's actually a very detailed 3D structure, as we can see in the upper part of this image here, where we have like filtered-like structures that start getting narrower and narrower until the fluid goes all through these structures and exits through Schlemm's canal. Several of the current glaucoma treatments that are available actually target this region in order to decrease intraocular pressure. However, due to its location in the eye, it's actually very hard to image this region. So one of the main problems is that even if we get light into this area, light returning back from the eye gets totally internally reflected and does not exit the eye, so we can't see it. To be able to overcome this, clinically, they use what is called a gonio lens, which is normally a plastic PMMA lens that couples to the front surface of the eye, to the cornea, through a layer of gonio gel, which has the same refractive index as water. So it lets light reflect off of an internal mirror and into the area that we want to image, but also allows light to come back out because we now don't have the interface between the cornea and air. The evaluation of glaucoma, however, is still limited due to the poor resolution of current gonioscopy, which is currently using a slit lamp. A slit lamp allows us to see this region. So we have the cornea up here in the upper part, the pupil here in the lower part, the iris, and in the middle lies the iridocorneal angle or the trabecular meshwork and neighboring structures. But this does not allow us to assess glaucoma or intraocular pressure. So what we wanted to do is actually combine gonioscopic imaging, as we see here, with commercially available optical coherence tomography devices. So this is currently used in the clinics as well, but in a different manner. So to be able to do that, we placed a button lens or a plano-convex lens on the anterior surface of this gonio lens to be able to focus light onto the trabecular meshwork. The gonio lens was placed on the eye coupled with gonio gel, and the pivot point of the OCT beam fell on the gonio mirror that enabled steering within the trabecular meshwork. The results we got are these images that you can see here. So if you look at this image, here's the iris, here's the cornea. So this would be an eye that is looking towards the right-hand side. We can see the trabecular meshwork, Schlemm's canal, and also corneal scleral bands, which are most likely due to a preferred orientation of collagen within this region of the eye. We compare that to what is obtained currently using OCT or optical coherence tomography, is that we aren't able to see the fine structures within trabecular meshwork or Schlemm's canal. This is one of the best images possible, and because it's imaging exteriorly without a gonio lens, we actually have a lot of light scattering in this region. So to be able to improve on this even further, I designed a new gonio lens, which improved the actual optical design of these lenses using airspace doublets and also changing the material that we're using. So enabled to reduce the amount of aberrations at that interface between the gonio lens and the eye. This was manufactured by a research center in Mexico, as we can see here, using a crystal, which was magnesium difluoride. And we are also working on making this much cheaper by 3D printing shells of gonio lenses that can be filled with the fluid that is index matched or has the same refractive index as our cornea, and hence reduce aberrations. The second part of my current project is working on the design and implementation of an imaging suite, which includes adaptive optics for scanning laser ophthalmoscopy, a line scan imaging, as well as optical coherence tomography. So this was allow us to get 3D cellular level resolution imaging of the drainage outflow pathway. So as I showed in the previous slide, but now at much higher resolution. But it will also allow us to work on the retinal vasculature or image the retinal vasculature so that we can actually do 3D tracking of individual red blood cells in retinal capillaries. As I explained the first example in the previous slide, so I'll concentrate on the second one. And the question is why we want to look at the retinal vasculature. In this case, I could dive into diabetic retinopathy, which is a leading cause of vision loss among working age adults in the developed world. It causes remodeling of the capillary vasculature and is associated to areas of reduced oxygen. So basically, as we can see here, there are lots of loops and turns and tight curves that don't allow blood to actually flow through them correctly or at the right level. So we want to look at flow in these regions. There will be areas that don't get blood flow at all and completely cut off. and also regions, as you can see up here, these little dots of ischemia or a lack of oxygen in that part of the retina. And this is what is affecting our vision. What we want to do with our design is actually be able to image the cellular level imaging of the retinal capillaries. We want to image the individual red blood cells, as we can see in these videos here. These white little dots are individual red blood cells that we can see. But however, we don't have enough speed to actually be able to track these. And it fails, these systems fail when we try to measure the velocity of them when we have tightly curved vessels. To be able to do that, we're using adaptive optics, which basically takes light that is aberrated coming back from the eye. It images the pupil plane onto an adaptive element that can correct this by measuring the aberrations first and working in closed loop with this adaptive element, which is normally a deformable mirror, to be able to correct that wavefront before we image it. So in this case, we can see a retinal image here, for example, with adaptive optics turned off, and this is the best possible compared to adaptive optics turned on, where we can actually count individual photoreceptors. So that is the level that we're working at. To be able to do that, I've designed a new system, and I'm currently building, that includes this scanning laser ophthalmoscope, which basically takes a tiny little spot focused on the retina and scans it so that we actually achieve an area of roughly about 2 degrees on the retina. But each spot is 2.5 microns on the retina, hence allowing us to get a very high resolution. But this is working at 30 hertz and hence not fast enough. The second part of this design is actually working on a line scan, which takes diffraction limited or very highly focused line on the retina, scans it only vertically. This allows us actually to work at 400 hertz frame rate and get the speed that we need to image very fast movements. The third part of this, and what will allow us to get 3D imaging, is actually the OCT. So this will still give us very high resolution in 3 dimensions, but all 3 of these are working with adaptive optics to improve on that. In this case, our OCT works with the same type of detector as a line scan, so these 2 will be working in conjunction. And this will allow us to the 2 systems to be inherently synchronized and reduce movements of the eye in order to align the 2 images. And because of the high speed, we'll be able to get a 2 hertz volume rate, which is very good for ocular purposes. So finally, the objective of this project was to build state-of-the-art adaptive optics imaging, including scanning laser ophthalmoscope, line scan, and optical coherence tomography. It will allow us to have a high lateral resolution to image the fine structures, but also allow us to have very high speed to measure small and fast movements within the tissue. We will be able to get the highest optical efficiency for the line scan ophthalmoscope, which has never been obtained previously, up to 98%. We'll be able to image the drainage system in 3D at cellular level resolution, which has also never been done before. We're going to track individual red blood cells through to be able to study the retinal flow, specifically for diabetic retinopathy. And actually, this system is very relevant clinically to be able to study many other diseases that have not been mentioned here, because we can image both the anterior part of the eye or the front of the eye, but also the back of the eye. So I'm going to end there, just saying thank you for all the funding I have got until now to work on these projects. My current group, which has been amazing, the group that I did my PhD with, Dr. Brian Bonson in Ireland, and also several of the institutes which I've visited previously throughout my research. Thank you very much. And if you have any questions, I'm happy to answer them.
So thank you very much, Alessandra. So if anyone have a question, please use the Q&A panel so we can go ahead and take those questions to Alessandra. So Alessandra, just a while the people is asking questions, in the experiment that you were doing with the DMD, the digital micromirror device, did you have any problem with the diffraction effects caused by the micromirror movement? And how did you solve that?
Yeah, so in this case, it wasn't a huge problem. It's separated out far enough that we could just cancel out the other diffraction orders and just use the zero order that we needed. But in other applications where you're very limited for the amount of light, for example, imaging, this is slightly a more troublesome problem. It can be overcome, and people have before, but it depends on the amount of light that you need to be for your application.